um, receptor protein interaction. So this virus, the piece that is exposed is just capsid. So this is a naked virus, right? Naked versus enveloped. On this side, right, it looks like this virus has an envelope and it has little spikes, right, that have native proteins. And that protein recognizes a receptor and then boom, comes inside, okay? So we need to have, it's a very specific interaction. And we mentioned how, again, we'll talk about it later, but what happens if you don't have a receptor that recognizes this protein, or if your receptor doesn't recognize this protein, then the virus can't come inside. Virus doesn't come inside, you don't see the infection, right? You don't see signs, symptoms, all that good stuff, okay? And we said we can blame our parents, right? Because it's genetics. So we can blame our parents based on this, okay? Also, for this interaction to occur, right, viruses, or typically these pathogens, like to infect a very particular area. So for example, COVID, right? COVID, that's a respiratory infection. So what happens if I eat COVID? Like if I ate the virus, would I get infection of COVID-19?
No.
No, right? Because it's gonna go to the stomach. Do I have the interaction with my cells in my stomach? No. Okay. So routes of entry is typically important, right? And what cells they interact with. Okay. What's another one? So like, let's say it's a GI, right? And it comes in and you inhale it. Is it going to, should it cause infection? No, it shouldn't, because it typically attacks GI, okay? So that's kind of the gist of it. So we want to make sure that it's attacking a very specific type of cell, right? And if not, no infection. Okay, this is talking about, again, how we name them. So we name them, we put them in families based on their genome. And then this is the example for HIV. So HIV belongs to the family Retroviridae, and it has the genus lentivirus, and the species is human immunodeficiency virus. So typically their name is going to tell us something about who it's infecting and what it's causing, okay? And then you can have different strains, so 1 or 2 or 3. Okay. Okay, and then the genome, right? So we'll talk about differences between viruses and how they replicate. So replication is going to look different, whether it's a DNA virus or if it's an RNA virus, okay? When we talk about RNA viruses, you can either have double-stranded RNA, so DS RNA, double-stranded RNA. You can also have single-stranded RNA viruses. When we think about single-stranded, so SS, there's 2 types. So there's positive RNA, also called, I think it's the sense strand. Okay, sense strand is the positive RNA. And then you have negative RNA. It's called the antisense. And antisense is the negative one. So this is kind of what that looks like. So double-stranded means that you have 2 strands of RNA, right? So RNA, RNA, it's double-stranded. Here are your single-stranded options. So if you have single-strand RNA and you're a positive strand, that means your RNA is in 5 to 3' direction. Okay. So positive, right? That's typically how we think of RNA, right? It's made in the 5 to 3' direction. So it's the positive one. If you have negative strand, single-stranded RNA, then your strand is 3 to 5. Okay, so that's the difference. And then the difference kicks in with when we think about how this cell is going to attack or begin taking over the host cell. So, the very first thing that a virus wants to do, right? It wants to replicate or make more genome, right? It needs more genome, right? If you're gonna make if you're gonna make a virion, you need more genome, right? And what else do you need? You need more proteins, right? Because this is gonna make your protein coat, right? Or your coat, right? So the two things that you need more genome, you need more protein coat. Okay, so this guy, if the virus comes in and it has positive sense, it can use this strand, and this strand can directly hop on a ribosome, boom, start making more proteins, right? So you can use the positive strand to make proteins. Perfect. Okay. But now, you need to do one more thing, right? What else does this guy need to do? Yes, we need more genome, right? So to make more virions, we need more. Here's kind of a tricky question. If I have a strand of RNA that's 5' to 3', how can I make more RNA? You actually have to make anticodon first. So you have to use this guy to make 3 to 5. So in other words, you're gonna make a negative strand. Okay? You're gonna make a negative strand from your positive. Okay? Then we can use this negative strand, and what can we make? More positive, right? Because the negative strand is a template for positive, right? Right? Because if we had, let's say, our positive strand was like A, U, C, G, G, A, U, A, right? That's our positive sense. We first need to make the negative sense. So what would the negative one be? U, right, I'm starting here, U, A, G, C, C, U, A, U. So that's my negative. Now I can use this negative to make a positive. And we can pump out, this will be our main template to make all the positives that we need to make the virion. Okay, so now we have our proteins. We need a template to make tons of positive strands of RNA. Okay? So we now have the two things we need to make more virions. Okay, if we look at our negative, okay, also call this antisense. Antisense, our negative strand. This guy, what can we use this guy to do? We can use it to make a positive, so we need to make a positive strand. And then what can we use this positive strand to do? Yep, now we can make some protein. Right. And then what else can we use this strand for? For the genome, because we need the positive to be a template to make more negative. So now we can use now we have our proteins and we have our genome. Right? In this instance, this is the template. In this instance, this was the template. So just a difference in what you need to make. Okay? And since we're talking about RNA, it's weird using the term genome because when we say genome, it could mean RNA or DNA. In this specific instance, we're making RNA from RNA, so that would just be transcription. We'll talk about when we make RNA from DNA, when we talk about DNA viruses, but transcription would just be RNA to RNA. Yes. Mm-hmm. Yes. So we're doing a lot of transcription. So that's what this is saying, right? So the positive sense, this can act as an RNA or like a messenger RNA and gets translated to a protein. Okay? But you need to make a negative sense to transcribe more positive. This guy, the negative sense acts as a template for RNA, so you can make your complementary positive strand RNA to then make more negative, and that positive is used to be translated into a protein. Either way, you need to make the opposite, right? If you started with the negative, you still need to make the positive. If you started with positive, you still need to make the negative. Okay? You need all parts for both. Questions? So either way, you need both. It's just what you started with. What was your next step? Okay. So, how do they multiply? So we're going to talk about first bacteriophages, and then we'll talk about animal viruses. So bacteriophages are viruses that only infect what kind of cells? Bacteria, yes. Okay, bacteriophages only infect bacteria. Okay. We're going to talk about lysogenic cycle and lytic cycle. Remember, we kind of talked about this a little bit already, right? Lysogenic, this is when a virus basically goes dormant, right? Like chickenpox or any type of herpes virus. They can go dormant and it can stay hidden in the DNA of your cells for a very long time until you get stressed out, Once you get stressed out, it jumps into lytic, right? Lytic, think just lysis. So this is, if something is in the lytic cycle, it's infecting and then destroying as it exits, okay? I know it says lysogenic and lytic under bacteriophage, but animal viruses can also be lysogenic or lytic cycle, okay? So both bacteriophages and animal viruses can be lysogenic or lytic. Okay, so first we're going to talk about bacteriophages. So phages are viruses that infect bacteria, right? They look kind of crazy. I don't need you to know T-even bacteriophages or lambda bacteriophage. I'm not going to ask you that. Yeah, just lytic versus lysogenic. I'm not going to talk about either of those two examples. Yes, we'll get into it. So, here. So, let's start here, right? Who's this guy? I got my nice little bacteriophage, right? It looks like a little alien. What shape is this? Complex. Right. And it's going to insert, let's say this is DNA, okay? It's going to insert its DNA, okay? That DNA gets incorporated into this plasmid, okay? So this is a recombinant cell now, right? Because it has new genes, okay? It's inside, then we're going to degrade everything, right? So that we can use these strands, messenger RNAs, if you will, right? Because we started with DNA. We could use that DNA to make RNA, right? As we typically do for all the other genes in here. We're going to make more viral genome, and we're going to make some proteins, okay? Once we have more genome and more proteins, we can assemble them, right? We can start to assemble our little virion, okay? And then once it exits, it just lyses the cell, okay? So this would be lytic, right? It just went in, replicated, and left, destroying the cell as it leaves. This would be lytic cycle. Okay. Within lytic cycle, these are the steps, right? So adsorption is kind of ugly. I'm not going to use adsorption. I'm going to say attachment, okay? So what does a virus have to do? It has to attach, okay? Then it's going to penetrate. Then it's going to biosynthesis, maturation, and release, okay? So attachment, right? How did the bacteriophage attach? All right, it attached with this little tail, right? That's the attached part. Okay. Then penetrate. So penetrate is how the information comes in, right? It got injected in. Okay. Phage actually uses lysosomes to open up the cell wall. Okay. Once that information comes in, then step 3 is biosynthesis, right? We produced more DNA and proteins. Okay. Maturation, we assembled the phage particles or virions, right? And then step 5, release. Okay. So what are the 5 steps? Attach, penetrate, biosynthesis, mature, and then release. Okay, so it's just 5 steps. And you can label them here too, right? Here we have, right, attach and then penetrate, kind of happening at the same time, right? Biosynthesis, right? Maturation, we're assembling them together, and then release. So this one shows it a little bit prettier, right? Here's our attachment. See, your book uses attachment and it uses adsorption, but adsorption is kind of ugly, so I'm just gonna attachment, or keep it. A is attachment, okay? So on the worksheet, when it says like step 1 or like A, if you think of like an acronym, so like A, P, B, M, R, attachment, right? So right there, it attaches. What's coming inside? DNA, right? So that's the penetration part, okay? Then we can replicate these contents, right? Make more genome, make more proteins. In this case, we need to make more capsid, we need to make more sheath, we need to make more tail, right? A couple more products to replicate. And then it's fully assembled, right? Once it's assembled, then it can release, okay? And as it releases, it is killing the cell. Questions. 5 steps to lytic cycle for this bacteriophage. Okay, we feel confident, Okay. The phage growth cycle. What I kind of care about is just understanding that there's a period of time, right, where when the virus first comes in, right, that information comes in, it's gonna take a while before that information is starting to get transcribed, translated, or replicated, right? Then it's gonna take a while before, as you start replicating the amount of virions that you're producing, you're still not really gonna feel sick, right? You're not gonna feel like sneezing, the itching, right? Vomit, whatever it is that you have. There's a latent period. So there's gonna be a period of time where your virus is replicating, but you don't feel anything. It's not until the viral load, right, is high that's when you start feeling sick, okay? So I'm not gonna test you so crazy on this graph. What I just want you to understand is that we start off not feeling sick, right? Because we're in latent, so you can't really feel the severity of your symptoms because there's not too many virions present. It's when your viral load gets really high is then when you start to feel sick, okay? That's it. Lysogenic, okay? So here's lysogenic. We're gonna start here, okay? So attach, penetrate, right? That genetic information gets inserted, right? So this is now a recombinant cell. But now we're gonna get fancy and we're gonna call it a prophage, okay? So this bacteria that obtained the genome from the virus, we call it a prophage. Before the virus, right? Because it's sitting there, okay? So this is a prophage. That prophage can sit. This prophage can divide, right? Can be divided dividing for many years, right? And then those cells continue to divide. So this is our latent stage, right? All these prophages that we're creating, just dormant, right? And then, right, if there is a time of stress, or, you know, if your cells are stressed out, if you're, right, sometimes it'd be like an exam, you get super stressed out, that's gonna cause this virus to say, ooh, I gotta go. This cell is stressed out. So this cell can jump into biosynthesis, right? Jump into the lytic cycle. The genome's already there, right? So all it has to do is start to replicate and make more proteins from the information, right? So we can make more genome, we can make more proteins, right? Assemble them, maturation, and then release, killing the cell. So again, we can be lysogenic for years, months, right? This is typically like herpes viruses, right? When you have herpes, you have it forever. Congratulations. Why? Because it gets incorporated, right? It becomes a prophage. Stays in our cells, right? Chickenpox. Chickenpox is a type of herpes virus, right? If you, depending on the type of herpes you have, right? There's like active phases. The active phase is when it's in lytic, right? When you have like a cold sore, it's in lytic, right? And then it can go dormant, cold sore goes away, and then you get stressed out, and then boom, cold sore comes back up, back to lytic. Okay. Questions? But still, same steps, same 5 steps how to happen. Okay. Now, animal viruses. Okay, so we're going to talk about DNA viruses, RNA viruses. My DNA virus, I'd probably just be like herpes. Herpes is a good one, right? RNA viruses, we'll talk about positive strand RNA viruses. My example for this is polio. So I'll give all polio. Okay. And then the negative single-stranded, this is, I think, influenza. And then we'll do double-stranded RNA and retroviruses. So it's going to be like HIV, right? HIV is a retrovirus. Okay. Steps. One more step. Okay. We learned 5 steps for the bacteriophages. Animal viruses, just one more step. Okay. We still need to attach. Okay. So we're gonna attach to our cells, right? And attachment can happen in 2 ways, right? Either it's the protein coat to our cell, or it's the spike to our cell, right? So attachment can vary based if it's envelope or naked. Okay? And then penetration, right? So how does it come in? You got 2 options. If you're an envelope, you come in through fusion, okay? If you are naked, you come in through endocytosis. Okay? We'll talk about that. Here's the fun one, okay? We're gonna add uncoding, okay? So this is like, if I'm gonna walk into my house and I touch the doorknob, that's attachment, right? I open the door, that's penetration. I'm coming inside, and as soon as I step inside, I take off my coat, right? That's like uncoding. So I'm exposing the DNA or RNA, right? Exposing the genome. That's the uncoding part. Okay. Then biosynthesis, right? Replicate genome, and then mature, so assemble your product, and then release. Okay. Releasing, you got two options. Okay, releasing, you either exit through exocytosis, aka you lyse the cell, okay, or buddying. Okay? So depending on if we have an envelope or no envelope is how you exit. If you have an envelope, it's gonna be buddying, right? Because for buddying, we wanna take a piece of the membrane with you, right? If we do exocytosis, we're just boom. We can just go through it. So just pop out. Pop out is gonna be naked, because naked doesn't need the membrane as it exits. Okay. I'll show you some pictures. Okay, but just to preface. Okay. So uncoating is that special step that you see in animal viruses, not in bacteriophages. Okay. So we can turn this in next week for some points. So how does a virus attach? Okay, so if you have an envelope, okay, here I have the receptor, I have enveloped virus with spikes, I get inside here's a receptor, and how do I come in? Fusion. Because remember, lipid with lipid and lipids fuse together, right? If I drop oil into water, the water doesn't mix with the oil, and then I add more oil, where does the oil go straight to? The other oil. So the oil likes to go together. So when you have an envelope, they just fuse. Boom. Okay? How did it attach? The spike, right? Spike if it has a spike. If it doesn't have a spike, then it's going to attach with the capsid. Okay? So depending which one you have, is how you're going to attach. Okay. Here's fusion, right? So I have my spike being recognized. Lipid, fuse together. Okay? Then, boom, uncoat. Right? Release the capsid. It's like taking its coat off, and there's free RNA. Okay? At the top, if you had a naked virus, so no envelope, how do we bring it in? Endocytosis. Right? So the cell is going to pinch around the virus and then bring it in. Okay? And notice it brings in tons of virus at a time. Okay? Here's a microscope picture that shows, that's pretty cool. So it's not alive. This is how they come in. There's look at that picture. It's so cool, right? Just like that. So typically, this route, you're taking in tons of virus at once, not just one. Okay? We do. That protein receptor, right, ligand receptor interaction says, oh, come on, trick or treat, welcome, right? Is us, right?
When you were talking about how it takes before we start feeling anything, did that make a difference between which one? Because if you end up listening more at once, then the fusion would feel like faster? Could you take it more?
I'm not 100% sure. You'd still have to replicate everything though.
Okay.
Yes. So the naked virus comes in through endocytosis. The enveloped virus comes in through fusion. So the attachment varies. You got two options for attachment. Okay. Then here's your uncoding. Okay. Uncoding 1 through 4 is just different ways to uncode. Okay. All I care about is that you know what uncoding is. Uncoding means that you're exposing the genetic material. That's it. These are just different ways to achieve that. I don't care that you know the different ways to physically achieve that. But then you gotta uncode, right? Expose that genetic material. Okay? No. I just need you to know that step and its purpose. The 4 different ways, I don't care too much about. Okay? I don't care about that. And this is unique to animals. Okay? Okay. Once we took the coat off, okay, expose the genetic material. Now I get to make more genome, right? So the way that you make more genome varies whether it's a DNA virus, RNA virus, right? Single-stranded positive, single-stranded negative, double-stranded RNA, or retrovirus. Okay. So depends, right? Okay. Here's where we can start a little bit, okay? Worksheet. The very first one, it says biosynthesis replication of DNA viruses, okay? So I would write next to it, example, herpes, right? Herpes is different. But if you need to visualize the virus in your head, I put herpes, okay? So, where is viral DNA replicated? Viral DNA is replicated for DNA viruses in the nucleus of the host cell. Okay? And then it says, does this process use host or viral enzymes? It can use host or viral enzymes. Then it says, where are viral capsid proteins made? It makes capsid proteins in the cytoplasm. What enzymes does it use? It uses host enzymes. And then it says, where in the host cell are new virions assembled? It says capsids migrate to the nucleus and you make your virions in the nucleus. Poxvirus is an exception. Because remember, this is my weird one, right? It doesn't have a capsid. Poxvirus, it does everything just in the cytoplasm. This is our weird one. Okay. Okay, we're gonna keep filling it out. Okay, so what are the general steps in DNA virus replication? Okay, what is the first step? What is A? Attachments. So write attachment. Okay. Since we're doing herpes, we have, is this an enveloped virus? So how did it attach? Ooh, that's the second one. Attach The recognition. Attachment. Could be either a spike, right? It's gonna be a spike or a protein. That's how you have attachment, right? Yeah, we're not there yet. We're not there yet. We're not there yet. No, not yet, not yet. That's attachment, right? Then you have, what is second step? Penetration, yes. Okay. And how did this virus penetrate since it has an envelope? Fusion, yes. So penetrate through fusion, right? What's the third step? Yup, uncoding. See, look, took its coat off. Expose the DNA right there. Beautiful. Coat off. Okay. Then the DNA is gonna go into the nucleus. Okay. Biosynthesis. In the nucleus, two things happen with the DNA. Okay. It says DNA is blank to make more viral DNA for packaging into new virions. What would we put? How would we fill in the blank? DNA is blank to make more DNA.
Replicated?
Replicated. You replicate DNA. Second thing that happens with the DNA in the nucleus, it says DNA is blank into mRNA, which then leaves the nucleus and goes into ribosomes in the cytoplasm. Transcribe.
Okay.
Beautiful. We know this, we just did this, right? That was exam 2. Okay. Okay. So it migrates to the cytoplasm. On the ribosomes, viral RNA is blank to make viral capsid proteins and viral enzymes.
Translated?
Translated, yes. Okay. Viral capsids, proteins, and enzymes go back to where?
Nucleus?
Yep, go back to the nucleus. In the blank, new viral DNA and new viral capsid proteins and enzymes are assembled into new virions. Yes, nucleus. Okay. Virions leave the nucleus and exit host cells by one of 2 mechanisms. For envelope, how did you leave? Budding. See? So as it exits, it's taking membrane with it. That's how it gets its new envelope. If it was naked, lyse. Just lysis.
That's how it gets
Also, another gross fact, right? Let's say this is COVID, right? Because COVID has an envelope. So my COVID virions that I made, it have my cell membrane, right? And then I sneeze, right? It's droplet, right? So if you come into attachment with a droplet, then the virions that went into, let's say, the person next to me, they have virions that have my cell membrane on it. Just to make it more fun, right? Super nice. Okay, so understanding what were the steps and where are we? Where do we do what, right? Come in, attach, penetrate, uncoat, straight to the nucleus, replicate DNA, transcribe, make messenger RNA. Messenger RNA goes outside to the cytoplasm. We translate, make proteins. Proteins go back to the nucleus. In the nucleus, you assemble, and then boom, exit. Okay. Questions for this one? we're gonna talk about some RNA, mhm. So it depends. So it can either be a spike, right? A spike if it has spike proteins on it, or it would be the capsid, right? If it has no envelope and no spikes. Mm-hmm. The protein? It protects the genetic material. Protein receptor interaction. Either it's the same you gotta interact both times. It's just what is it? Is it a piece of envelope or is it the capsid? Because the capsid is what's exposed. If you have, here's my previous example, right? If you have a positive strand RNA, you can use that positive strand to directly make proteins, but then you need to make your negative to make more positive. If you have negative, we can use that, we need to use that negative to make a positive in order to make more proteins, in order to make more negative strand. Okay. We're gonna use, for replication of RNA virus, and it says polio in parentheses. Polio is a positive single-stranded RNA. Okay. So it's a positive single-stranded RNA. So this is my example. Okay. So where is viral RNA replicated?
Cytoplasm.
Cytoplasm. Okay. Where are new viral capsid proteins and enzymes made?
Cytoplasm.
Cytoplasm. Okay. We have a positive strand, okay? Step one, what can we do? Oh, hold on, before step one, it's looking for attachment. So step one is attach, okay? Polio is a naked virus. So polio is gonna attach. And then what's step two? We attach, and then what do we do? Penetrate, okay? Penetrate. It's naked, so how does it come in? Oh. Yeah, it's naked, so how does it come in? Not fusion. Endocytosis. Okay. Step three, uncoat. Right? And then D, okay, now we're here. Okay, biosynthesis. Two things happen. Okay, that positive strand RNA acts as a messenger RNA and is directly blanked by host cell ribosomes to make new viral capsid protein and enzymes. What is the blank? Start with the T.
Translated.
Translated, yes. Okay. Positive strand is blinked into negative strand RNA to make more positive strand viral RNA for packaging. What is our blink? Transcribe. Okay. Maturation, new positive strand viral RNA and new viral capsid proteins and enzymes are packaged into new virions in the cytoplasm. Okay, release new virions. This is a naked virus. how is it gonna exit? Lysis or budding?
Lysis.
Lysis. Boom. Negative sense. Negative sense. I don't have special words to go with this, but, okay, we're going to attach, right, penetrate, uncoat, then we can use the negative strand to do what? We gotta transcribe it into the positive, and then the positive we can use to translate some proteins and transcribe more negative sense. Then you have maturation, right, assemble, and then release. Okay, last one. And then that's it. Okay. And this is just a picture of how you have release budding. Okay. With budding, if you have slow budding, you're not really killing the cell. But if you have tons of budding happening at once, then you could totally like kill a cell by doing that. But the goal wasn't to kill, but you could still potentially kill it if you're like going crazy with the budding. But yeah, sorry for that. Okay, HIV. Okay, HIV, when it comes to replication, it's a little different. It's a retrovirus. It's pretty cool. Okay. If this is right, we have to attach, penetrate, blah blah, all that. But if we're just looking at replication, okay, it comes in as RNA, and it comes with special proteins in it, okay? One of the proteins being reverse transcriptase. So reverse transcriptase is a protein, what do you think it does? It's called reverse transcriptase. Yes, so it can actually make DNA from RNA. So you're bringing in RNA, you're bringing in reverse transcriptase, right? That's unique to the virus. So what's it gonna do? It makes DNA from your RNA, and then that DNA can get incorporated into the host cell. Congratulations, you now have this forever, okay? At any point, if it turns into lytic, right, we have biosynthesis, then you can make more RNA from your DNA, and you can make reverse transcriptase now, right? And you can make more enzymes and more proteins that this virus needs before it exits. Okay? So this is what's special about the retroviruses. Retroviruses can go backwards. So they make DNA from RNA. Okay. So D, replication of retroviruses, aka HIV. Step 1. Here we go. What's step 1? Attach. Okay. HIV is special. Okay. HIV has an envelope and it has spikes. The spike that I care about is GP120. Okay. It's the only spike I'll ever ask you about. HIV has GP120. Our receptor recognizes GP120. Okay, so it attaches through that spike, GP120. Once it attaches, what's step 2? Penetrate. How is it going to penetrate? It's enveloped, so how does it penetrate? Fusion. Okay, fusion. And then what's step C? uncoat, take the coat off, okay? Now we're gonna get into biosynthesis, okay? So, the positive strand RNA is blanked into DNA. Reverse transcribed. I know it's a small, but reverse transcribed. Okay. Using the viral enzyme, reverse transcriptase. Beautiful. Okay. Newly formed double-stranded viral DNA inserts into the cell chromosome, becoming a, so this time instead of a prophage, we call it a provirus, right? Prophage was for bacteriophage, provirus is for our cell, animal cell. Okay? This is a recombinant cell, but provirus. Okay? Animal proviruses cannot come out of the host chromosome like bacteriophages can. Okay, so I do that differently. We got it forever. That's what that means. Okay. Step 2. While inserted in the host chromosome, the viral DNA can be blanked into new viral mRNA. Transcribed, right? You can transcribe DNA to make our new viral messenger RNA. Viral messenger RNA can be used to make more viral RNA to package into new virions. Okay. B. Viral messenger RNA can also be blanked to make new viral capsid proteins. Translated. Step 5, maturation, right? New viral RNA and new viral capsid proteins can be assembled into new virions. Okay. Now, exiting the cell, okay? How does HIV exit? It has an envelope, so budding. Yep.
Can you repeat that last part?
How does it exit? Budding.
Before that.
B, viral mRNA can also be translated
Okay.
to make new viral capsid proteins. It's a form of exocytosis. Questions? How do we feel about this? It's just a lot, but we did it together. And I gave you a worksheet. So will this come back? Yeah. Okay. When's our next quiz? Okay, so I'll probably ask you about parasites, right? So be ready for some parasites finally. And weird and viruses. So parasites and viruses. Okay? It's coming up. How do you feel about this worksheet though? Better? And then again, each one has a slide that goes with it. So just practice knowing what is happening where, depending on what virus. Okay? And then those steps that differ between envelopes versus naked. Okay? You feel good about that? Knowing that polio is positive, naked. That's the only example I've given you. So the three guys that I talked about here, where is polio, HIV, and then the DNA. Which in my mind, I just like to say herpes. Because it didn't make sense to me. Right. Chickenpox. Right, that's a DNA virus. So, but yes, those are the three that we mainly went over. But technically there was 4. And this one came with that special protein, reverse transcriptase. Right, do we have reverse transcriptase? No, we don't have that. So it had to come with that already, right? If it didn't have that, can it do this? What happens if it didn't have reverse transcriptase? Yeah, it can do any of this, right? Okay. And then, again, weird thing, right? We said there's about 1 to 2% of the population that's essentially immune to HIV because their receptors, so HIV specifically likes to target T cells, T helper cells, and macrophages. So the specific receptor is CCR5 and CCR4. So this one, CCR++, this means that you have a normal functioning CCR+ gene from mom and from dad, right? Your 2 chromosomes. Because you got 2 copies of everything. So you are susceptible. This one, you have CCR5 normal and then mutated at 32. So if you have half, then you are susceptible but may clinically progress less rapidly. Okay, so, and then this one you have mutation on both. So mom and dad. So that's that rare 1 to 2% highly resistant to HIV. So weird, right? No. I hear GP120. I won't ask you CCR5. Questions? Yeah, that's the GP stands for glycoprotein. So that's that protein on this, that's the spike. Okay, we're gonna end it there, because I wanna go home and take my medicine and wake up a new person tomorrow. I'm trying to knock myself out and see if I am new, if I wake up new. It wasn't the flu, it was more like a cold. And it started because the kids got sick, and the kids got me sick.
Yeah.
I wasn't as bad as them,
Is
but
there what do you feel more comfortable
I
with?
had like a headache, the congestion is what gives me the headache.
Yeah,
And then
I'm down with that. Yeah, I'm down if you want to do
now I'm a
the
vampire,
model
don't
part.
expose any
Take over
light
the
to me,
report
I'm
part.
all sensitive to the
The
light,
report.
and I'm just
Like
We have to make, you mean like a report, or do we actually have to do it?
I believe. Let me look at it because I thought we had to do a report with
it's
it.
about that time.
Model on the board.
Once I'm not sick, then I'm gonna go get the flu shot. I like to get the flu shot.
Maybe I'm tripping.
We can ask tomorrow and then.
Yeah, maybe I'm
Figure
tripping
like
because
a
you know how
plan
there's
out.
some they look like a science project and then they wrote like
Yeah.
their thing.
Yeah, that's like the board though, right?
Oh, is it? So is that?
It's on your-
Okay, so then the board is like the report part.
Yeah.
Okay.
Maybe like this, they have like cut out.
Yeah. And then the models actually.
Yeah.
The model. I think the board.
Okay.
Yeah. But I mean, either way, like I'm probably gonna ask you questions about the word, and then
Yeah,
you'll probably
that's fine, because I can work out.
yeah, but yeah, for sure. But yeah, I'll look at it this week. This week I work like all week, but
So,
next week